Hello, everyone. Thank you for joining us for the 2021 Microbial Pathogenesis and Alzheimer's Disease Grant webinar. In this webinar, the IDSA Foundation will share insights into the grant application process and tips for making sure your submission stands out. My name is Evelyn Morales, and I'm the program manager at the IDSA Foundation. I'm responsible for growing and managing the foundation's many programs, including the Microbial Pathogenesis and Alzheimer's Disease Grant. The Microbial Pathogenesis and Alzheimer's Disease Grant supports research that suggests evidence that an infectious agent or microbial community is correlated to Alzheimer's. The grant promotes novel research in the field of microbial triggers for the disease. This year, the foundation is thrilled to announce an increase in grant funding. We're providing up to $1.7 million in total awarded grants and new grant levels that expand the reach of this opportunity. Today, we'll be talking to two past grant awardees about their experience and how the grant funding opportunity impacted their research. I'm happy to introduce Dr. Laura Cox and Dr. Colette Siwes Bentley. Thank you both so much for joining us today. I'd like to start by asking each of you to tell us a bit about your area of research and why you decided to apply for the Microbial Pathogenesis and Alzheimer's Disease Grant.
So our research has focused on exploring the role of the conserved microbial polysaccharide named PNAG that is expressed on a broad range of pathogens and looking at its disease pathogenesis. That's where we started. Part of this research led us to realize that the tissues that were thought to be sterile, meaning no viable, culturable, or PCRable microbial material was detected. Yet when we looked using our PNAG-specific antibodies at these tissues, we could still detect PNAG-positive microbial material. So we thought, well, clearly this must be trapped in these tissues. When we looked at human autopsy sections from Alzheimer's, Parkinson's brains, and compared them to age and sex-matched, non-neurologically affected brains, we also detected PNAG-positive material. So we wondered whether if this material could be contributing to the neuropathology in Alzheimer's disease. I'm an infectious disease cell biologist, so this was right up my alley. Where did we learn about the grant? Well, we saw it through the RFA posted on the IDSA website. And I guess we just decided to throw our hat in the ring because we were so far out of the Alzheimer's field. What could we lose?
Thank you so much for sharing that, Dr. Siwes Bentley. It's always fascinating hearing about each awardee's research because there are so many ways you can approach the topic that this grant covers. Along that line, Dr. Cox, can you share what your research focuses on and how you learned about the grant?
I first learned about the grant also from the IDSA website and how they promoted it in the field. I'm a clinical microbiologist by training, and so I really came up in the field looking at sort of overt pathogenesis, so Staph aureus and infections, or different soft tissue infections, and I one of the things that always struck me is, of course, that in early life and in elderly, we're much more at risk for infectious diseases. And so my work through my PhD started looking at how the early life microbiome could lead into obesity. And while I was doing my PhD, I had a friend that was looking at metabolism and Alzheimer's disease. And we said, wait a minute, if metabolism can be affected by microbes, and if metabolism is important for Alzheimer's disease, maybe microbes are actually important for Alzheimer's disease. So we did this study to look at how the microbiome changed in aging in these Alzheimer's mice, and we saw that there was a tremendous overgrowth of a bacteria called Bacteroides fragilis. And as an infectious disease microbiologist, as a clinical microbiologist, I was really familiar with B. frag. It's a common cause of infectious diseases like liver abscesses and all sorts of infections, but it's also extremely common in the gut. And it can live as a commensal microbe. And we know that there are good Bacteroides that make nice things for us, like certain vitamins. They can produce GABA, but there are also B. frag strains that have pathogenicity factors. And some of these pathogenicity factors can contribute to chronic diseases like IBD and even colon cancer. And so that made us ask, could B. frag actually be driving some of these changes in the brain. And so this was sort of a surprise to us that we found in our data. And so we grew Bacteroides fragilis, we administered it to a model of Alzheimer's disease, and we saw indeed that Bacteroides fragilis could promote plaque production in the brain. And so that was really surprising to us, and we said we have to learn more. Now, because of how common B. frag is, the world over, most people, many people have Bacteroides in their gut. And so we don't think that it's reasonable to go ahead and try to knock out Bacteroides from all individuals. So we want to understand, are there specific pathogenicity factors? Are there strain-specific things that we could screen for that are really contributing to the Alzheimer's disease pathogenesis? And if this is true, then what we could do is develop some PCR screening methods that are commonly used in clinical diagnostics. And so we want to understand more about the biology, and we want to understand how to sort of track, prevent, and then maybe intervene if this bacteria is contributing to plaque production in the brain.
Clear that both of you are making great strides within your research. When you learned about this grant opportunity, what made you decide your research was a good fit?
So the link between specific microbes and Alzheimer's disease has never really made sense to us, as it seems to us that there are multiple pathogens that are contributing to the Alzheimer's pathology. PNAG being a broadly expressed polysaccharide on bacteria, fungi, and some parasites made it a really interesting target to detect the presence of a wide variety of pathogens. So not limiting the etiology of Alzheimer's disease to a single pathogen. We thought this was a great fit and would allow us to explore many pathways.
And I was really excited about this grant opportunity because I had been working in sort of more general connections between the gut microbiota and Alzheimer's disease with immune modulation. But I saw this grant as an opportunity to really do some nice microbial epidemiology and start to discover specific strains. Now, we do know with many diseases, you can think about GI illness, there's a wide variety of microbes that can contribute to that. But we still can develop screening strategies for the most common pathogens. And so I thought this was a really great opportunity to really understand the microbiology and bacterial factors that could be underlying these processes.
Dr. Cox, as a 2020 grant recipient, you have gone through the application process most recently. How was your experience applying for the grant?
So I found that the online portal was very simple and straightforward. I did listen to the webinar, that was helpful. And, there was the combination of the processes of the scientific proposal, plus thinking about what it meant for the community, for the public. And I thought it was a well-rounded grant application, and I found it fairly easy to use the submission portal.
Dr. Saiwaz Bentley, you received grant funding from the 2018 cycle, the very first grant cycle. Can you tell us about your experience throughout the process?
I thought it was pretty straightforward and easy. I can't really remember so far back, but the only thing I will say is if at any time when I had problems or issues, especially with COVID, reaching out to Evelyn was just amazing. She's prompt, she's rapid and concise, and really helped solve a number of issues that I had had. So that would be my advice. If you've got questions, ping Evelyn.
Thank you, Dr. Saiwaz Bentley. I'm here as a resource and I'm happy to help however possible. We want to make our grant awardee and applicant experience as smoothly as possible. I know COVID-19 drastically impacted both of your research projects. Did you find that the IDSA Foundation was flexible during that time period?
For me, yes.
Yeah. Yes, absolutely. So we were able to make progress even during COVID, but some of the things shifted a little bit. There was less subject recruitment because of concerns of COVID actually being transmitted in stool samples. But we were able to establish some in vitro models and continue our work. And the IDSA was really helpful during this period.
Dr. Saiwaz Bentley, as an inaugural grant awardee, can you share how the grant funding has allowed you to progress your research over the last few years?
So as with Laura, we were also able to do some work during the COVID lockdown, especially with the 2 cohorts of mice. we generated an amazing set of data from this study that we have been able to use as preliminary data for additional funding opportunities. And it's also helped us to connect with other established Alzheimer's investigators that have helped us look at our data with different eyes. Clearly, it's helped us connect with Laura. And I think that the sharing of information, the ability to reach out into a new field has certainly become a lot easier having the data that we have on hand. So I'm hoping that our publication will come out once we have the samples processed, which was the biggest holdup from COVID. And yeah, so obviously apply for the IDSA funding for this year. And I'm hoping that we can, because it's only a 1-year grant, and most of the type of research that we do, the mouse model takes more than 1 year. So that is one thing that I think needs to be looked at clearly, depending on what kind of research the IDSA wants for this program. But I'm hoping to mine deeper into the tissues that we have obtained from the mice, the 2 mouse cohorts that we've been studying, and to pair that with our collaborators at Tufts, who are working in a 3D stem cell culture model to look directly at what does this microbial pathogen material containing PNAG actually do, and can we see that both in the mice that have gone through the study already, the vaccination studies already, and can we then match that with human tissue?
Dr. Cox, you have progressed your research as well. How has the Microbial Pathogenesis and Alzheimer's Disease grant impacted your research?
So the IDSA funding was really instrumental in allowing us to start to search for these new microbial strains from Alzheimer's patients. And in the last year, we've been able to isolate 25 new Bacteroides strains from both healthy controls and Alzheimer's patients. And using in vitro models, we're starting to find differential activities related to amyloid beta uptake using in vitro models. And then we're breeding up our mice post-COVID. Well, it's not really post-COVID, but we're starting to breed up our animal colonies so that we can start to test the effect of these strains in vivo. This really helped us, launch into a new area, and also provided pilot data to apply for additional awards. So those applications are still pending at the moment. I would say the other thing that I'm very thankful for the IDSA is raising the level of awareness for this really important factor. We know that there are many different environmental factors that may play a role in Alzheimer's disease. And given the growing public health burden, it's really important to intervene. And since the IDSA has started this funding opportunity a number of years ago, many other funding agencies are now starting to get interested in this and support this area. And I think we're going to need a lot of scientists at the table exploring this area. And I think the IDSA has really helped lead the charge to get this point out and raise awareness.
It sounds like both of you have enjoyed the experience and have made great progress because of it. As previously mentioned, there will be an increased grant funding this year with up to $1.7 million in total awarded grants. Individual grants range from $30,000 to $250,000, expanding the scope of eligible applicants from fellows and early career investigators to established researchers. What would you say to researchers across all experience levels to encourage them to apply?
Absolutely. So if you think you have a new exciting idea, take the risk, start to put it together. It's really important. There are many different aspects of these types of investigations, whether you're an Alzheimer's researcher, whether you're a microbiologist, whether you're a bioinformatician. The first step is really put together a good team. And we have many great researchers at our center. And so I would say, put together your team and really just go for it. And if you're an early career investigator, try to make your question simple, feasible, addressable. It's a big challenge to try to do anything in Alzheimer's disease in a single year. But definitely, I would say go for it. It's a great opportunity.
So as I think, based on Laura's comments, this is a developing field, and sometimes when we go into things, we don't really know what we're going to see. And when we when we get the outcome, it opens so many more doors and so many more questions in the field. And I therefore think this is a great platform to take what we have, not only to publish the existing data, but then to mine that data further with different questions, perhaps bringing in other people's in different fields that we hadn't previously thought were connected. One of the things that I feel very strongly about is that we have so much to learn about this disease, and that having curious people from different unconnected fields work on the problems at hand can only bring fresh perspectives, but also allows us to re-examine old dogmas with fresh eyes and new tools. It can only be good for making advances in Alzheimer's disease. And I think that this IDSA platform allows that to actually happen.
Your work is dedicated to digging deeper into Alzheimer's disease microbial roots. Why is this research important? Why should we invest in this area?
So we know that the greatest risk factor for Alzheimer's disease is aging. And if we're all lucky, we will all age. We also know that even though we've identified familial mutations, they account for 10% of the disease. So 90% has some sort of environmental trigger. We can change things in our lifestyle, but maybe there are other things going on that we should pay attention to. We know that in aging, we're at increased risk for infectious diseases. And so as there's immunosenescence and also inflammation and aging, we need to think about strategies to prevent the growth of microbes that could be causing damage to the brain. And so we think that understanding specific pathways, specific mechanisms will allow us to have a more targeted approach. And I think those are the tools that we really need. We need to understand this biology so we can get in there with molecular diagnostics. These could eventually be implemented in a healthcare setting in a very cost-effective way. We've seen with the COVID outbreak how quickly you can do a PCR for a specific pathogen. And this has been used, to look for pathogenicity factors, in staph and other things like that. So if we can really identify the specific processes, then we can start to actually intervene with some knowledge. And I think that's the missing link, is we need to understand the players and then find a way to put information in the clinician's hands to promote healthy brain aging. The grant opportunity really helped us give resources and time to start to look for the microbes in Alzheimer's patients and start to find this connection so that we could actually, in a way, have the fulfillment of Koch's postulates, which is to find the organism in a disease versus healthy state, give it to animals, reproduce the disease, re-isolate it from that animal. And so we'd really like to do classic microbiology experiments to demonstrate the role of that pathogen in the disease. And we couldn't do it unless we had the funds to say, okay, we're going to now do these very focused studies and generate isolates coming directly from the Alzheimer's patient. So that really helped us address something that's not out there in culture collections. We needed to have our own strains. And then again, having the IDSA grant kind of helped me connect in with Dr. Cywis Bentley and others at BWH that are studying this area that really have a lot of unique expertise. And so that really helped launch this investigation and also connect me with others in the field.
So what I think is really beautiful about this is you actually have two investigators that are completely on the opposite spectrums. Laura's looking at an individual pathogen, and we're looking at a broad marker, literally just a tag for microbial presence in the brain of Alzheimer's patients, and looking at whether just the presence of microbes can result in inflammation and contribute to Alzheimer's disease. So the IDSA granting agency has enabled two very, very different approaches, two completely opposite spectrum approaches to study the same pathology. And, you know, we might both be right. I think that this syndrome of Alzheimer's disease and these age-related diseases is not going to be as simple as we wish it were, to have only one therapy to solve all the problems. It's going to be a connected team approach to actually make any headway with these diseases. So I think that that really embodies what the IDSA's grant was about, bringing this kind of research together.
Thank you, Dr. Cox and Dr. Siwaz Bentley, for sharing your experience today and providing valuable insight about the grant. If you tune in today, we hope you'll visit our website, and that is idsafoundation.org/ALZ-research-grant for answers to frequently asked questions and more information. We hope you will apply, and we look forward to reviewing your application.